This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 372, recorded on January 15th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
We have a completely clouded up sky here in New York City today. And the temperature is 8 degrees Celsius with rain in the forecast for the weekend.
Pretty much the same here. It's high overcast, temperatures 5 Celsius, and they're saying it's going to pour rain much of the weekend.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. We also have 100% cloud cover. It's currently 39 Fahrenheit, 4 degrees Celsius. That said it's going to start raining in 4 minutes. But for the rest of the weekend, I don't see that there's much rain in the forecast.
So last Friday, you were having an impending snowstorm. Did that happen?
Let's see. Did it come as snow? We did get snow, but it wasn't Friday. Yeah, we got snow later. Saturday, yeah.
All right. Also joining us from North Central Florida, Rich Condit.
Uh-oh.
I was going to say sounding crisp and clear.
I was muted
on his new router.
Here I am. Hi, everybody.
Doesn't get any crisper or clearer than that.
You were muted. Did you mute yourself?
Yes, I did.
All right.
Because I have a respiratory infection that I've been fighting for some time, so I need to sort of gurgle and stuff in the background. 71 degrees Fahrenheit, clear blue skies. 22 Celsius. That's about the name of that tune.
All right. And Rich has been now home for 2 TWiVs.
That's correct.
That's the measure of being at home, how many TWiVs you've been at home for.
Yeah.
This episode of TWiV is brought to you by the 32nd Clinical Virology Symposium. You can now send in your abstracts to go to this meeting. This is taking place May 19th through the 22nd. In Daytona Beach, Florida. And the deadline for abstract submission is March 17th. And you can talk about your research on rapid viral diagnosis, the clinical course of viral infections, and ways to prevent and treat virus infections. There will be over 1,000 researchers and primary care physicians at this meeting. So if you get sick, you can have a physician treat you.
And then afterwards, you can ride your Harley down the beach.
That's right. You have a Harley? Do you have a Harley, Alan?
No.
No, you have a Wovel.
No interest in a Harley.
You don't even have a Wovel.
No, I have a broken Wovel.
Yeah. Yes. Well, if you're interested in this meeting, you should go to the website. It's asm.org/CVS2016. We thank ASM for their support of TWiV. And remember, if you'd like to advertise on TWiV, TWIP, TWIM, TWiV-o, Urban Agriculture, send a note to sponsor at microbe.tv. We reach tens of thousands of people who love science and love the weather. So think about that. You want to sell a weather thermometer. It's the right audience. We have some follow-up. Philip Miner, the senior author on the polio vaccine paper that we did last week, wrote, "Happy New Year. Andy sent me the TWiV thing." That's really funny, which I thought was pretty good. I am less used to reasonable discussions than I used to be, or would like to be, to tell you the truth. However, this business about licensing needing a protective efficacy trial so no change is possible is all very well, but Japan has licensed Sabin IPV, and so has China, and the issues are the same, i.e. no protective efficacy trial. WHO have guidelines on what you need in a clinical trial, although one does not have to pay attention, of course. WHO are also pushing Sabin IPV pretty hard with a view to getting its manufacture out into developing countries, or at least new ones, to reduce the cost of IPV. I accept that FDA will cut up rough and that it is a bit of a fiddle, but I think new vaccines like Sabin IPV will appear, and if them, why not S19, I ask you? Kathy provided a translation for
will cut up rough means to become very angry. And it is a bit of a fiddle means time wasting or trifling behavior, nonsense, triviality.
In my years of knowing Philip, which goes way back to the late 70s, early 80s, he's very colorful in his speech, and it gets into his writing as well, as you can see here.
These are British phrases, aren't they?
Yeah, he's
Yes.
from the UK.
Oh, yes.
So
We last week we had Wigs on the Green, too, in Downton Abbey.
Right.
If anybody hadn't checked that out.
Yes.
So I wrote to Philip, I think your S19 is lovely, but I didn't know that China and Japan had approved Sabin IPV, that is making IPV with Sabin strains. Of course, their approach is to get around that because the Sabin strains can revert. And I don't know. And so I said, will the UK use this? So he wrote back, I think the UK might. They did meningitis C vaccine with only immunogenicity data, although to be fair, that was backed up a year after introduction by data that showed it worked like magic in preventing disease. HPV was put on the market on the basis of its effect on cervical
cervical intraepithelial neoplasia. Right, CIN.
Cervical intraepithelial neoplasia. The I was
Correct.
catching me. Rather than cervical cancer, which it is intended to deal with, I suppose you can argue that cervical cancer is always preceded by CIN, but that all cases of CIN do not lead on to cervical cancer. So if you prevent CN, you prevent cancer, but it is still indirect. Given that humoral immunity protects against polio, ... I am sure that these arguments will only convince certain regulators, but there are political reasons for them to be convinced, i.e. the need for lots of cheap IPV. Do you think a new manufacturer using the current strains could get a license without any efficacy data? This gets a bit tricky. So the issue here is, I've been in lots of trials where these things come up. If you had an issue and someone sued, the fact that you went to market without efficacy data would be brought up in court, and you would
Sure.
pay dearly for that. So, I think the U.S. will not do that.
So, what are the correlates of protection in polio? Is it known?
Yeah, it's antibodies, for sure.
So antibody does it.
Yeah.
So it seems to me that if you can prove that a certain vaccine stimulates enough circulating antibody to do the deed, that ought to be enough.
It should be enough, yeah.
Ideally, yes. But in the FDA's case, I mean, they're the ones who are going to get called out if anything goes wrong. And certainly right now, they're looking at a situation where there are approved, established vaccines and now combination vaccines that roll the IPV directly into other vaccines. It's all very straightforward and by American standards cheap. There's absolutely no motivation for them to go out on a limb and approve something new.
Except that now the whole issue here is that, in particular, if we want to ramp up IPV production so that it can be used globally, we're talking about manufacturing facilities that are growing huge quantities of wild-type virus in an environment that we're now trying to keep free of wild-type virus. So the situation has changed a little.
It has changed a little, but from the standpoint of the FDA, they only have jurisdiction in the U.S. where this is not an issue. And for the overseas market, I'm pretty sure that certainly with an approval in China of a Sabin-based IPV, that's undoubtedly going to be a lot cheaper than whatever the American companies are turning out. And that is probably going to become the backbone of, or if India does something like this, that would then become the backbone of developing country IPV distribution. And as I said on the last episode, we're going to end up with a two-tiered system where you get one type of vaccine in the developed world, another type of vaccine in the poor countries, which is already what we have, but it's going to become more so.
I don't know that anyone knows what will happen in the U.S. because most of our IPV currently comes from non-U.S. manufacturers. I'm not aware of any U.S. manufacturer of IPV, for example, Condit up in Canada.
Right.
Now, as pressure to get rid of wild-type strains increases the type 2 this year. I don't know what's going to happen to manufacturer. They might switch to Sabin, but again, the FDA will have to approve it without any efficacy data.
And first, they'll have to decide that the U.S. is actually going to ban the use of the wild-type virus.
It may be that they do a phase 1 and see the antibody responses, Rich,
right.
and then say, hey, these are pretty similar to what we get with wild IPV. Let's go with this. I just don't know. I don't think anyone can say at this point.
But I think that approval is going to have to be rolled into the whole discussion about whether we're going to ban the wild-type virus.
Interesting situation.
Yeah.
It is.
In particular, if you have foreign manufacturers who may be under different regulatory issues manufacturing vaccine for the U.S. That throws another wrinkle into the whole thing. That's very interesting.
Yeah. All right, Kathy, can you take the next one?
Sure. Fernando writes, "Hi, Twivers. I was just listening to 371 where you kindly discussed my letter. Later in the show, Kathy seemed to be having trouble with the Google Ngram searches I had suggested. I wonder if she was including the quotes. I put the quotes in to separate the queries from the body of the letter text. They should not be entered in the Ngram viewer search box. Best, Fernando." So I went back and tried again. I'm not sure if I had the quotes in, but in fact, it works fine without the quotes. So if you have, for instance, *ADJ virus, then you get uses of adjective before virus, and then you can do virus *_noun. But one time I did miscopy it, and I had one quotation mark in front of virus, and that corrected to quotation mark space virus *_noun, and it gave a different result. So that was interesting too. But if you have two quotes in there, Google just comes back and says, this doesn't work. So that's probably what I was doing wrong last week.
Okay. Very clear. Alan, could you take the next one?
Sure. Catherine writes, "Dear TWiV, thanks for the great podcast. I always enjoy listening to it. Just a few points about the Tasmanian devil segment. First, devils are not the only carnivore native to Tasmania. Quolls are also found there. I think I actually knew that from an episode of the Krat Brothers from that my daughter had watched. But
did Dixon say that they were the only carnivore in Tasmania?
He wondered.
Uh-huh.
He wondered. And I don't think, I'm not sure where we came down on that.
Okay.
Second, I believe that the tumorous cell that's responsible, that's the cause of devil facial tumor, is thought to have originally been a Schwann cell.
Quolls.
Quolls. They're sort of a, they look a little rat-like, but I'm sure they're not in any way related.
I think they're kind of cute.
They are
spotted fur and
till they bite you.
Yeah.
Right.
Schwann cell, by the way, is a type of glial cell. It, helps keep neurons in shape.
Mm-hmm.
Yeah, and people get benign tumors of the Schwann cells, vestibular schwannomas, also known as acoustic neuromas.
So this is if it's a Schwann cell, then it somehow got outside of the nerve and started and got into the mouth, because that's how they're transmitted, by biting, right?
It got out of the nervous system. It surrounds the nerve in the first place. That was what you meant.
Well, they're also components of peripheral nerves, or is it only central nervous system?
Not sure. Don't remember that much.
Good question.
Neurology.
I think I've seen Schwann on peripheral nerves, in which case it just needs to get out of the nerve. And I mean, it's in your tissues, although you can imagine in your mouth you may have some as well, so it's not too far to go. Let's look it up.
I am right now.
Find out where Schwann cells are located. Yeah, glia of the peripheral nervous system.
Glia of the peripheral nervous system, yes.
So maybe there are some in the mouth area, or not too far from there. Just because if it were in the brain, then it could still get out, I suppose.
Sure.
Okay, Rich Condit.
Basel writes, "Dear TWiV team, I hope this email makes it in time, as I wanted to follow up on the cell type of devil facial tumor disease. The same group from the paper you discussed, GM Woods, had previously characterized these tumors as of Schwann cell origin, primarily based on the expression of periaxin, which is a marker they used in this paper." Okay, here's the link. He gives a link to it. "You've heard it many times, but thank you a ton for this wonderful podcast. Kind regards, Basel. P.S. I like that the authors are from Tasmania. It can't be any more authentic than this. University of Tasmania, Tasmania, Australia. P.S. That would be, second P.S. Shout out to all veterinary scientists and pathologists out there who contribute to such great science.
Indeed.
Rats. This link requires some kind of...
No, not rats.
Again. Oh, okay.
It's not working.
Walls. This link needs some kind of...
it's he probably put it in from the university. Yeah. All right. Let's try and find it. Kathy, can you take the next one? A little out of sequence here.
Josephine?
Yeah.
Writes, Dear TWiV-ologists, what a lovely surprise this morning when I checked out TWiV for today's download. You used George's Take Aim at West Nile painting for this week's podcast illustration. Thank you for your positive comments. I've asked my husband what the little upside-down umbrella thingy is in the cartouche. His best guess, this many years later, is that it is a stylized bulrush in a marsh, another mosquito breeding area. The marsh, not the bulrush. The weather here has improved considerably over the last 24 hours. Yesterday morning was a chilly minus 26 Celsius, a beautiful winter wonderland with everything covered in hoarfrost. This morning, the temperature reached 0 Celsius, so much more pleasant for outdoor activities. Best regards, Josephine. Nice.
Does anyone remember where Josephine...
I was wondering myself.
I don't think that you included it in last week's email.
Let's see. I think, did she say the weather last time? No, she didn't. So wherever you are, it's winter, I guess.
Someplace
I've
real winter.
Yeah. Okay.
I've got this paper about the Schwann cell origin.
Okay. You can put it in the link.
It's a science paper from 2010. I'll give the PubMed link.
Okay.
What's I could probably find. I put a link to George's website in last week's show notes, and that probably on his webpage probably has where it is. But anyway, it doesn't matter. All right, the next series are from Anthony.
Yes.
Who wrote that in the recent TWiV you remarked that human gene modification is part of evolution. That's what I said. Yeah, that brought to mind this by Chardin. It's a quote, with the discovery of genes, it appears that we shall soon be able to control the mechanism of organic heredity. The dream upon which human research obscurely feeds is fundamentally that of mastering, beyond all atomic or molecular affinities, the ultimate energy of which all other energies are merely servants, and thus, by grasping the very mainspring of evolution, seizing the tiller of the world. That's from The Phenomenon of Man, written in 1955 by Pierre Teilhard de Chardin. And he gives a link to that. And then last Sunday, I finished reading the New York Times, and I tweeted, every Sunday after reading the Times, I lament the absence of enough science, the field that makes our lives better. And Fake de Pommier replied to that. The arts and snarky columnists make our lives better, too. Weather, chat, and snark are key to the popularity of TWiV, etc. Okay. To which I replied, make our lives better. Not like science does. But the problem is science doesn't sell. Fashion and arts do. It's all about money. And, I don't care that the Times has all this stuff, and I just think they need more science. That's all. And then Anthony replied, that's certainly true for you and should be true for all. Honesty is something that lights up your podcasts. Unfortunately, even my very limited perspective shows the flaw of deep deceit that runs through society has reached science. Every other paper on the sex life of some invertebrate proudly proclaims the coming cure of Alzheimer's and or autism. Might this twisting of truth be an effect of the grant application process? Well, everyone likes to get their stuff out there. Well, not everyone. Many, many people like to get publicity. In fact, we have university publicity offices to tout the value of what's going on. And yeah, it's all about money to a certain extent. But money runs the world, right? Sure. I think the Times need more science because I know they have Science Tuesday, and that's great. But, Sunday, okay, they have other stuff, and I know people love it. But people need to know science, too. This is a case where the Times has done very careful surveys, and they know the science articles don't do as well as the fashion or the human interest. I understand that, but sometimes you need a loss leader for good purposes, and science deserves that.
Right, and there's some people that don't get the Tuesday edition. They only get the Sunday edition.
Yeah, that's right.
The problem really, though, is that the Times can no longer afford a loss leader because the entire paper is the entire newspaper industry is kind of a loss leader now.
They're still publishing fashion and arts. And
they are,
blah, blah,
because
blah, blah,
those
blah.
you can still sell it. You can still sell ad space in those, and you just
okay.
The economics of it make it really tough.
I'm just lamenting. That's what I did.
I also lament. Yes, this is my life.
There was actually a good article in the magazine section about a lawyer who chased down some toxins that were being dumped in Virginia, but it's more law and controversy than science, right?
Yeah.
I would, in that magazine, the section is wonderful articles. There should be one nice science article, and people will read it. Get them used to it. I'm sorry. Also,
well, you guys got to write it.
Yeah, I'm not a good writer for the Times. The other people, Alan has to do that. Dave Toller, other people.
Yep, they hire me.
Rebecca Skloot. All right, then Anthony also wrote, and this was brought up last time. I read his tweet, and he said, I would be proud to have written, access is not just a convenience or courtesy, it is essential. Without access, it's not science, it's authority. But I did not. It actually was a tweet by the anthropologist John Hawkes, and he gives a link to that. All right, last night yesterday I got an email from, I think, Health Map or one of these infectious disease maps saying, the Ebola outbreak is over. WHO has declared the most recent outbreak is over in Liberia. All chains of transmission have been stopped in West Africa. And I went over to ProMed Mail, and there's an article. And then the follow-up article is new case in Sierra Leone.
Yeah, it was on the news this morning.
Yep.
Well, and I tweeted the WHO release on the ending of the epidemic, which I think from the headline down, they talked about how this is tenuous.
Yeah, of course.
We're declaring the end of the epidemic, but we got to keep an eye on this because this could very well break out again. And then, of course, immediately you get news of a body testing positive.
Yep. Hours after WHO declared the outbreak over. So we'll see if this goes anywhere.
I presume they won't sit back, that they'll stay vigilant.
I certainly hope, yes.
Yes, I'm sure they will.
Just for kicks, I looked up the Ebola situation reports, and the last one was January 6th, so they have discontinued those as well, which makes perfect sense.
Right. Yeah, it was this health map was sending me, weekly, and they said, we're not going to do this anymore. We still have this page, but we're not going to send weekly updates, so... All right, we have a couple of snippets. I want to make these true snippets because we have two of them. And the first one has to do with...
And because our main paper is probably going to take some digging.
Yeah, we'll keep that down to 10 minutes, too.
Yeah, right. Good luck.
Influenza C Virus. This is a paper published in MBio and March, April 2014. So it's a little old, but this has come up recently, and I went back to the original article. And it is entitled Characterization of a Novel Influenza Virus in Cattle and Swine, Proposal for a New Genus in the Orthomyxoviridae Family. And the authors are House, Collin, Liu, Wang, Sheng, Liu, Wang, Nelson, and Lee. From the Newport Laboratories in Minnesota, South Dakota State University, and Shandong Academy of Agricultural Sciences, and also someone here at Columbia, whom I don't know.
And because it's in MBio, that means it's open access.
Yes.
I just want to say, go ahead.
Okay. Quick thing about typography. I was reading this article and the next two that we're going to talk about, which are also open access, available on PubMed Central. And both of those are from Science Advances. But the MBio format, I was reading it on my iPad, and it is much easier to deal with the full text format for MBio on the iPad than it is to deal with the PubMed Central format. So if anybody from PubMed Central is listening, please go over to MBio and copy what they're doing with full text for iPads. Because the figures, if you're in PubMed Central and you bring up one of the figures in the paper, it's just useless. You can't bring it up to a large enough size, so I just went to the PDF.
Okay. This paper has to do with Influenza C Virus, which I don't think we've ever dealt with very much here on TWiV.
Really doesn't come up much.
Doesn't come up much. I mean, remember, the influenza, Orthomyxoviridae, which is the family that includes the influenza viruses. There are 3 genera of influenza viruses, A, B, and C. And, we vaccinated against A and B. And C are typically considered to cause. In fact, most people are seropositive after a certain age, but they're not very serious, and so a vaccine has not been warranted. However, I've always had a warm place in my heart for Influenza C virus because a good part of my thesis was on these viruses. And I found one of those articles. Well, they're still there, of course, on PubMed. They don't go anywhere. This is from no, that's the wrong paper. Oh, that's the wrong paper. Here we go. This was from, it's got to be the 80s. No, 1979.
Wow.
Isolation of Influenza C Virus recombinants. So I was the first to show that these viruses reassorted, not really recombinants, but reassortants. This is way before I was fussy with words. 1979. And I was looking at the paper, and we did a cool experiment. So we took influenza C virus, and I had run some RNA gels to show that the genome was segmented, and there were only 7 segments as opposed to A and B, which have 8, because one of the segments encodes a protein that has both the functions of the hemagglutinin and the neuraminidase, the surface glycoproteins. And then I UV inactivated 2 different strains of influenza C virus. And individually, when you infect cells, they're not infectious. But if you mix the UV inactivated virus stocks and then infect, you get good infectivity in the viruses that are coming out, or reassortants. Isn't that cool?
That is cool.
That was Peter's idea, I'm sure.
Wait a minute. I don't get it. Okay, because does that mean that
you inactivate one?
You're not
UV
inactivating every segment.
Exactly.
Okay.
Exactly. Dr. Condit still has it.
Yes.
I'm working on it. I'm struggling.
Despite a year of retirement.
Yes. I'm struggling.
I know. I thought it was cool. And then we took those viruses, we labeled the RNAs, we ran them on gels, but the 2 parents didn't have distinguishable RNA mobility patterns. So then I cut out each individual segment and did RNase T1 2-dimensional maps to show the reassortants. Yeah, a lot of P32. We used to get 50 millicuries a week. Anyway.
So did you label those viruses in vivo?
Yes.
Did you label those viruses in vivo?
Yeah.
Did you label them
in cells, yeah.
You labeled them in the cells with inorganic phosphate?
Yeah. That's
man.
Cheapest way to go.
Isolated them and ran the gels and cut out the gels. What a mess. Yeah, you were swimming in nukes.
No, after you dump off the media, you dump off a huge portion of the inorganic phosphate. And
still a lot of nukes.
It's what we did in the Holland lab, too. We'd get 50 millicuries a week.
Well, for my thesis, which was a whole lot later than that, I was using huge quantities of orthophosphate to label virus.
Anyway, brought back memories. Sorry for the digression there. But,
and the number one hit in 1979 was My Sharona.
My Sharona. I like that song. It's a good song. So this paper, they have essentially... they essentially have identified what they think is a new strain of influenza virus. They call it Influenza D. Although I went over to ICTV, even though the title of this says Proposal for a New Genus, when you have such a proposal, you got to go to ICTV and make a proposal. And I looked, and there isn't one there.
Oh.
So it's too bad.
They don't call it D anywhere. They just say that they propose it should be a new genus.
Yeah, but there are more recent papers now that call it influenza virus D.
Oh, okay.
In fact, I saw one of those, and that's what made me go back to this original.
They should get their paperwork in for that.
They should get their paperwork in. So
they probably have it in, don't we think?
I don't know. The most recent ones were from the end of 2015, and this paper is from 2014. So, it should be in already. So, Jens, if you're listening, do you know what's going on with this influenza D virus proposal? Anyway, when I did my thesis on influenza C, it was believed to be an exclusively human virus, but in the 80s, there were reports of isolation, first in 1981 from pigs in China, and then from pigs in the U.S. in 2011. And these are viruses quite similar to human isolates of influenza C virus, but not cross-reactivity with human strains. And serological studies subsequently found that 19 and 10% of pigs in the UK and Japan are seropositive to human influenza C virus. And 10% of pigs had titers to an isolate from a pig called C. Oklahoma, C/OK. So it looks like these viruses are circulating in animals. They wanted to find the reservoir, so they did scans of cows, cattle,
right.
and they found the virus in cattle as well.
So these were respiratory cattle that had developed respiratory illness, and the samples were sent in from that.
So in this paper, they've done some sequencing and other studies which show that the sequences diverged, the splicing patterns of the RNAs are different, and so that's part of the reason why they'd like to make it an influenza D virus. So
how was this first picked up and identified as C?
Well, it was first isolated from pigs in China in 1981. Okay. It was clearly Influenza C virus because they sequenced it. Genetic analysis of the Chinese swine IC.
Well, it would be closest to Influenza C.
Closest to C.
Okay, yeah. because now we're calling it potentially a new genus.
Right.
Okay, and depending from the phylogenetics that they present, whether it's closest to C or something else, kind of depends on what sequence, on what segment you sequence. And according to the antibody cross-reactivity, it's not cross-reactive with human C,
that's
right?
Correct. That's correct.
Okay. So
they have a figure here where they do the phylogeny with all the 7 segments. And you can see that they're all related. These bovine strains, they're on the same branch as some human strains. In fact, the ones I worked with years ago. So they all have a common ancestor. That's clear from the phylogeny.
Right. Oh, I just saw right at the end of the introduction, they refer to it. They say
influenza D virus.
Influenza. Right. This would be the type species of influenza D virus, but it's part of a kind of a ambiguous statement.
Yeah. So C. Oklahoma 2011. That's the American isolate of C in swine. That's the type species, and they say that's influenza D.
But it's probably not a native virus of swine, which is why they were looking at cows in the first place. It doesn't seem to really circulate much in pigs, and you find some of it, but that doesn't seem like really its reservoir host. So then the evidence from this paper suggests that maybe cows are the real host for it.
So they got samples from cows from 6 different states, 8 samples, which is 18% were positive for this particular virus by PCR. And then they isolated virus in cell cultures.
Excuse me, what percent were positive?
18.
18% out of their first sample of cattle, right?
Yeah. 45 samples and 8 were positive by PCR. And then they recovered the virus by infecting human. This is really strange. They use human rectal tumor cells. I don't know why. Maybe that's the only one that worked, but it seems like a strange place to be looking for this. But anyway, now
it's almost like they pulled that cell line out of their butt or something.
Wow. All right, so that's basically the paper. They have some sequencing and some they do reassortment. Yeah, this is the other reason.
Right.
One of the ways you define A, B, and C is that they don't reassort with each other. So A only reassorts with A, B only with B, and C only with C. And they tried reassorting the bovine with the human, and they don't reassort.
Which matches the classical biological definition of a species. Can it mate with?
Yeah.
So the pig virus and the cow virus will reassort with each other, and the human C viruses will reassort with each other, but the animal C viruses will not reassort with the human C viruses. So that justifies, among other things, putting it as a different genus.
I do note that their reassortment was not done the way we did it. They brute forced it. They just picked lots of plaques and sequenced the individual segments to see where they were coming from. I think it would have been easier.
That's the way you do it now.
Yeah, but
looking back at this phylogenetics, it's like there's only like 6 out of the 7 segments have sequences that cluster close to, though clearly distinct from, C viruses, and one of the polymerase genes clusters closer to A and B than to C. So I can see how if you did sequence, you would conclude initially that it was a C virus.
Right. They also have a nice figure here where they look at 8 different herds in different places, right? And you can see there's substantial. There are isolates in all these different herds, so it's suggesting that it's relatively widespread. And then
what I couldn't get out of that figure is what fraction of animals. They give a mean HI titer over the whole herd, but I couldn't figure out what fraction of animals were infected. So the only data we have on that is from that first sampling they did where it was 18%.
Yep.
Right.
And then they did the immunological cross-reactivity that Rich referred to, and these do not cross-react with the human strain. So those are some of the reasons why they would like to make this Influenza D virus. And whether or not these viruses infect people, we don't know. But nobody pays much attention to Influenza C. But I guess this is a good thing to keep an eye out for.
Sure.
So
go ahead. I was just going to say, in the end of their discussion, they talk about this COK virus. The ability to infect and transmit in ferrets suggests this pathogen has a potential to cause disease in humans. Did we see that in this paper? The ferret data, or was it data not shown, or I missed it?
That came as a surprise to me when I got to the discussion.
Yeah, I assumed it was some other study, but they didn't even reference it, so I don't know.
Right.
But I noted in that paragraph that they think the virus spilled from cattle to swine, and they say, maybe it could go into humans as well. So we'll see. Anyway, I thought people should know about Influenza D virus.
Sure.
If in fact the ITC, what is it?
ICTV.
I can't get it right. ITCV accepts it, right? They have to accept it. But it seems like there's good evidence for it. All right. So the other snippet is a paper, also open access. It's published in Science. What's the name of this?
Science Advances.
Science Advances. Damn. Why don't they have it on the front page? Research article. They don't have the name of the damn journal. Damn it.
When you look at it in PubMed Central, it's right at the top.
Damn it, Janet.
If you print it out, it's at the bottom of the page.
Well, I don't print.
I know.
Let's see here. Oh, on the right side, downloaded from advances.sciencemag.org. Okay. Anyway, Science Advances is Science's open access offering, or one of them. They probably have more than one. And this caught my eye. It's called Recurrent DNA Virus Domestication Leading to Different Parasite Virulence Strategies. And this is from France, Montpellier.
Authors.
A bunch of authors. First author is Apolline Pichon. Oh, nice name, Apolline. And the last author is Anne-Nathalie Volkoff. And they are from Montpellier, Tours, and where else? Okay.
From France.
Different places in France. Oh. This is to do with the Wasp endogenous viruses that we love to talk about.
Yes.
Pallidnaviruses.
Except these aren't actually pallidnaviruses.
Yeah, but we've talked about pallidnas before.
Yeah, we've talked about pallidnas before. That's how we sort of think of them as a group.
These are echinoviruses. And so pallidnaviruses, remember, these are carried by many parasitic wasps, and they include the brachoviruses and the ichnoviruses, and these infect braconid and ichpneumonid wasps, respectively, and they package DNAs and put them into the caterpillar and along with the wasp eggs, so that when the wasp egg hatches, the gene products are made from the viral genome, and it helps it immunosuppresses the caterpillar's response and allows the wasp to grow. So they're really important for that.
So these are both these are both parasitic wasps with the life cycle that was the inspiration for the movie Alien.
Trying to make a long story short, all of this stuff comes from these wasps having been basically endogenized by a virus eons ago, a Nudi virus, which is like a baculovirus. And so the Nudi virus got into them, and its genome became integrated so that it's in and in various ways scattered throughout the genome of the wasp. So it's a part now of the wasp genome.
That's what you mean by endogenized.
Yes,
right?
and in these different lines of parasitic wasps, the expression of those endogenized ancient virus genes manifests in various fashions. The one that we've most commonly talked about is encapsulating another set of wasp DNAs that encode immunomodulatory genes in virus capsid proteins to be delivered along with the eggs and compromise the immune system of the parasitized caterpillar. And this is about a wrinkle on that theme.
So these ichnovirus carrying wasps, which are called Venturia canescens, now they actually deliver virus-like particles to the caterpillar, and these VLPs don't have any viral DNA in them. They have proteins in them, right? They are proteins wrapped in a viral envelope, basically, and in this paper they want to know how that works. Now, my question for you, Rich Condit, do all ichnemoid wasps do this, VLPs, or is it just the Venturia canescens? All right, so that I don't know.
I don't know.
Okay, so it may be that not all of the ichnemoids do this. Maybe
the two really new things for me here are, first, that these virus-like particles are these membranous things rather than the proteinaceous capsids that we've seen before, number one. And number two, that they're delivering protein rather than DNA.
Right.
Okay, so fascinating, because the overarching theme is the same, but and it's from eons ago. Still, it looks like a Nudi virus, as we'll get to, but after the probably initial endogenization, the way it ultimately plays out is very different.
Right.
Wow.
So you've got both of these wasps got infected, possibly in separate events a long time ago, by this virus. And then rather than getting sick with the virus or whatever, they domesticated the virus, as these authors put it, and put it to their own purposes. But they then, in these two different species, put it to completely different used completely different approaches for that.
So basically what they did in this paper is they looked in the tissues that are producing these VLPs. They're produced in calyx cells, and then the particles move into the oviduct, where they then move out along with the eggs. And they did mRNA sequencing analysis and said what transcripts are made. And they found transcripts from viral genes, 7 viral genes encoding viral envelope proteins. All right, so these are being transcribed in the tissue that's making these viral virus-like particles. So then they also looked in the tissue by transmission electron microscopy. You can see the virus-like particles being assembled. And then they took these virus-like particles and they did mass spectrometry to identify the proteins that are in them. And they identified 26 these are Nudi viral gene products, including all of the envelope proteins encoded by those 7 genes that we just mentioned. They found the mRNAs for, and they found virulence proteins of wasp origin that are thought to be given into the caterpillar to ensure immune protection. So it's basically that part shows you that the VLPs are wasp virulence proteins wrapped in Nudi viral envelopes. So the envelope proteins the envelope proteins drive the budding of these things.
So one of the interesting things is that if you look up the structure of a Nudi virus, okay, and this is pretty unusual for DNA viruses, it seems to me. It's a double-stranded DNA-containing virus, and in the virion, the DNA is wrapped in protein in a nucleocapsid-type structure, and that whole thing, I guess a helical, the way it's pictured, at least on Viral Zone, is it's like a helical nucleocapsid structure, and that whole thing is enveloped in a membrane that's a lipid bilayer with virus proteins in it. And interestingly, what's absent from all of these transcripts and stuff is the nucleocapsid proteins. Right. So, and which makes sense for doing what this thing is doing. You've gotten rid of the nucleocapsid protein so that you no longer encapsulate any DNA or anything like that. You're left with just the membrane structure, which is now directed to encapsulate these virulence proteins. Unbelievable.
So those genes left years ago.
Yeah.
And they developed this strategy emerged. What I find interesting, so the driving of budding of the particles by envelope proteins, that's cool. That happens with other viruses. But how did the Wasp proteins get packaged into them?
It's weird.
Right? How did that happen?
Initially by accident, I'd guess.
Yeah, but I would guess you need specific sequences to interact with
I was thinking the same thing. If you once they have a closer look at this, they'll probably find that the Wasp genes that get packaged have evolved over time somehow to be specifically packaged into these VLPs. After all, in the case of the polydnaviruses where you're packaging DNA, the Wasp genes, the Wasp virulence genes that get packaged actually are modified in terms of their context in their genome so that they're specifically amplified out of the genome and specifically packaged and delivered, and in that case actually integrate into the genome of the cells in the caterpillar and express proteins. So there's significant evolution of the virulence genes in the Wasp to accommodate them to this whole system.
So do you think?
And I would expect the same of the proteins.
Do you think that the Wasp proteins are interacting with parts of the viral glycoproteins that are sticking through the membrane? Probably, right? To direct
Sure,
them.
yeah, something like that.
So you could do some nice in cell culture experiments to show that and to identify the interacting amino acids and change them and so forth. So you could imagine some nice experiments coming out of this.
I could imagine also that there might be some interesting work to do on the host.
What you got in mind?
Well, I mean, this viral mechanism, this rather baroque mechanism for delivering these immunosuppressive molecules from the Wasp, there almost has to be the complement of that going on in the caterpillar that it's trying to infect. I mean, this type of mechanism obviously evolved because there was some selective pressure from the host to have this type of mechanism. And I think you might be able to use this to get at something about the immune system in the caterpillar.
Yep.
So I don't know whether to call this convergent or divergent evolution, because you start off millions of years ago with a similar, if not a similar initiating event, which is a wasp being endogenized by a Nudi virus. And then these things take different pathways. So that's kind of divergent in a way. And yet the overall effect in the end, that is protection of the eggs in the parasitized host, is the same. So that's kind of convergent. It's
right.
Yeah. These were
bizarre.
separate. These are separate infections, they think, right? So
yeah.
So the ichnovirus, which is the VLP ancestor, was one infection. They lost the nucleocapsid protein and the ability to package DNA. And then separately, and more recently, they think, 100 million years ago, the Nudi viruses, the beta Nudi viruses, in fact, and those maintain the DNA packaging and so forth. So there's two different ways to achieve the same end, which is to suppress the caterpillar so your eggs survive, right?
I'll bet
Also
there's
makes
more
me
out
wonder.
there.
Yeah, it also makes me wonder how many more examples of viral domestication we're going to find.
Yeah, just grab wasps and start sequencing them. You could do that.
And why limit it to wasps?
Sure. Sequence the Earth.
Yes.
They also, by the way, sequenced the V-kinesins genome to show that, in fact, these wasp virulence proteins, the viral genes are integrated in there, and they're part of the genetic material of the wasp, which is pretty cool.
One of the things that I think about as I read this, because I've been following this literature for, 30 years, 25 years, is how different it has become technologically. I remember reading papers very early on in this where they're still dissecting ovaries out of wasps and getting stuff out of dissected ovaries. But before, it was getting from hundreds of wasps minute quantities of RNA to make cDNA from to try and clone things. And now with the sequencing techniques being so sensitive, it's so much easier to get so much more information.
Yeah. It's not free though. Remember, you got to pay for
No,
it.
not
Sequencing is costs.
And I'm glad that at least the French are willing to pay for science, basic science of this type. Though some of this gets done in the States as well.
Yeah,
Sure.
Our friend Mike Strand and others.
yeah.
Mm-hmm.
So this is, viruses of immunity of viruses in parasitize wasps. You can see this being a Golden Fleece Award on the floor of Congress, right?
Mm-hmm.
And yet it's just absolutely fascinating basic science.
Yeah. Okay, that's why we need more science in the Times so that people will understand. You can imagine
actually,
this would make a cool article, wouldn't it?
Absolutely.
Actually, they have a cool sentence at the end of here. Recent decades have seen the investment of considerable amounts of energy and money into the development of liposomes, or virus-like particles, for delivering molecules of therapeutic interest to target cells. We show here that natural production of such vectors was selected in parasitic wasp lineages long before the first primates evolved and their descendants were able to develop biotechnologies. Implied in that is that this basic stuff may have a biotechnological application down the road as well.
And also nature does things way before we do.
Yeah.
Including gain of function.
Yeah, including gain of function.
Mm-hmm.
I think we've hit all the high points there.
Yeah, that was a good sentence. I like that.
Bingo.
All right. Then our final paper, our main paper, is also in Science Advances, so you can take a look at this as well as you're listening, but not if you're driving, of course.
No.
Cellular defense against latent colonization foiled by human cytomegalovirus UL138 protein by Lee Albright, Lee Jacobs, and Kalita from the University of Wisconsin-Madison. And you know one of these people, Rich Condit?
I do indeed. Derek Jacobs, one of the middle authors, got his PhD from Dave Bloom's lab here at UF. I was on his committee.
Good to see.
So good to see he's doing well.
So this is all
and
about.
Actually, while I'm at it,
Yep.
I want to shout out to Dave Bloom Lab in general. This whole thing about chromatin modifications having a role in herpes latency had its birth in Dave's lab.
Very nice.
One of his students got interested in this. It was, as I understand it, a student-generated project, and said, let's have a look at this. So good for Dave.
I thought this would be a nice follow-up to the herpes simplex virus paper we did a few weeks ago, where we also showed, in that case, emerging from latency in response to neuronal stress involves a pathway to chromatin modification, right? So this is a different virus. This is human cytomegalovirus, and it looks at a different aspect. But again, it's chromatin modification, which has to be altered in order to get herpes viruses out of latency and get them to make infectious particles. So this is cytomegalovirus, which is a beta herpes virus, different part of the herpes viridae family. And this is like most of the other herpes viruses. This is a virus that infects almost all humans at some point in your life and becomes latent, stops making virus particles, and then reactivates periodically to make particles and spread the infections to others. This is a big problem for people who get organ transplants because you have latent CMV and you get immunosuppressed so you don't reject the organ, and you can get very nasty CMV infections. It's also a virus that crosses the placenta and can get into the developing fetus and cause serious congenital defects.
Maybe you said this, but very widespread in the human population. So
Yes.
large fraction of the population has it. And if you're immunocompetent and all that kind of stuff, it's pretty good. It's when under conditions of immune compromise that it's a real problem.
It's something like 80% of us are walking around with this thing, depending on whose estimates you
Yeah, as I understand it, a significant fraction of your immune system is dedicated to keeping this thing under wraps as you get to be elderly like Dixon.
Hey, he's not here to defend himself.
That's
Even
okay.
better.
I have a way to summarize kind of the take-home message of the paper. I think that's a segue from what we were just saying in the previous paper about nature and biotechnology. In the end of the discussion, they say, our immune system learned long ago what scientists only recently discovered. Latent reservoirs are seeded too rapidly for innate or adaptive immune responses to effectively control. Thus, faster-acting intrinsic defenses may be especially effective against a process that happens quickly. So we're going to hear about that
perfect.
intrinsic.
That's perfect.
Yes.
Yes, that's a good segue because this is about intrinsic defenses, which are in cells. They protect individual cells. And we've talked about some of the proteins. Unfortunately, they're called restriction factors, which is really unfortunate.
Very, very confusing.
Well, they're proteins, first of all, and I don't like the restrictions. They're part of the intrinsic defenses. We have talked about apobex, the proteins that deaminate HIV genomes to cause mutations, and TRIM5 alpha, SAMHD1, etc. Now, the important one for this paper is a protein with the lovely name of DAX, DAXX, which stands for death-associated protein, I believe, correct?
Mm-hmm. Death-associated protein 6.
Protein 6, DAX, DAXX. And these proteins inhibit the immediate early transcription of herpes viruses. So this is a countermeasure to blocking viral propagation. And remember, immediate early transcription is what you need to turn on in order to go from latency to productive infection. So the idea here is, as Kathy said, this happens quickly, so you don't have time. No way the adaptive response is even going to have time to play a role. And even the innate takes a few hours to kick in. So you need something that's already there. And these, quote, restriction factors, unquote, are there in cells. They don't need to be turned on so they can deal with these viruses.
And this immediate early gene, IE1, is sort of the central player in all of this because it's a major transactivator of early gene transcription, I guess. And so if you're going to go down a lytic pathway, you express that gene and it turns on early genes and you're rolling towards a lytic pathway. If you suppress expression of that gene, you're on your way to latency.
Right.
So the activity of that gene is a critical switch in this whole thing.
So the virus, HCMV, when it infects a cell, it includes a viral protein in the virus particle called PP71. It's part of what's called the tegument, part of the virus particle that has a number of viral proteins in it. As the virus gets into the cell and the genome is delivered to the nucleus, this protein goes into the nucleus as well, and it causes degradation of DAX, because DAX is a countermeasure to viral immediate early transcription. Right? So if you don't have a PP71, then DAX will basically silence immediate early transcription, and it does so by inducing a repressive chromatin structure. Remember, we talked about this last time about how methylation of histones can cause repression of transcription, or I don't think we talked about acetylation last time, but if you take acetyl groups off histones, it turns off transcription as well.
Right. So it's DAX is if there nothing interfered with DAX, it would come out and ball up the viral DNA around immediate early genes so that they couldn't transcribe.
So the cool thing here is that the virus is actually helped by DAX in a way, because DAX will silence the viral genome, and that allows for a latent infection. So in some ways, the virus evolution has taken this into account, and it's not really a bad thing to have DAX silencing it.
Well, and in fact, whatever, depending on the cell type that it infects. Right. So if it infects a terminally differentiated cell, then the virus will send, well, let me get the sequence right on this. The PP71 does go to the nucleus.
Mm-hmm.
Correct.
And that stops DAX, causes immediate early 1 expression, or permits it, And then that sets off the whole chain for a lytic infection. So the virus replicates and lyses the cell.
Correct.
And that, for example, is in a fibroblast.
In a fibroblast.
Terminally differentiated, but like in a fibroblast.
Right. So if it infects one of the cells where it would normally establish a latent infection,
like an incompletely differentiated cell, like a myeloid.
Like a myeloid lineage cell.
Some sort of myeloid stem cell.
Right. Then PB71 stays in the cytoplasm.
Mm-hmm.
And this is because of contextual differences within the cell, as I understand it.
Yep.
And because PP71 stays in the cytoplasm, DAX remains active, balls up immediate early genes, immediate early 1, on the chromatin, and stops the lytic infection, which allows the virus to establish the latent infection.
Right. Now, there's an important piece of information you need to have before we talk a little bit about the results, and that is there is a compound called valproic acid. This is an inhibitor of histone deacetylases. Now remember, histone, if you take acetyl groups off of chromatin, you turn off transcription. Okay? So a histone deacetylase will turn off transcription. You guys want to disagree?
No, that's correct.
Right.
I've got a cheat sheet here, okay, that I wrote down to keep track of this because it's so confusing.
Acetyl off, transcription off. Methyl on, transcription off. It's the opposite for the two modifications.
Correct.
So this acetylase inhibitor, so a histone deacetylase will turn off transcription because it takes acetyl groups off. If you inhibit the histone deacetylase, it will turn on transcription.
But you inhibited the inhibitor.
Right. So DAX is working at the level of histone deacetylases. And so if you throw in an inhibitor of the deacetylase, DAX can no longer silence, and you get around it. And they use that in a lot of experiments. Right? Okay.
Good.
And also here there are effects of... so that they make a nice statement in the introduction, which I like. This is a unique example where a virus does not neutralize an intrinsic defense against productive infection, that's DAX, but actually uses it in certain cell types to promote latency and persistence. That's what I was kind of talking about before, but not so eloquently, right? And then we're also going to be dealing with methylation of histone.
Right, because it's much easier to understand when you actually anthropomorphize it.
They said actually uses it. I don't like that, but notice I did mention.
Yeah, I mean, in order to make this comprehensible, you have to.
Yeah, there's a lot of anthropomorphism that goes on in this paper, and I agree with Alan. It's almost necessary. The best way to. But
listen, guys, I don't want to be saying this all the time, but the authors of this paper haven't probably even thought about anthropomorphizing because they've been brought up like all other people trained like I was to anthropomorphize, and only if you write a textbook with Jane Flint do you learn not to do that. So I'm not going to harp on that, but that's fine. The other modification is methylation of histones, which we talked about in that last paper on herpes simplex virus, right? The neuronal pathways leading to alteration of the histone. And I like the way they address it in this paper. They call it writing the modifications or erasing the modifications on the histones. That's a really cute way of doing it. So latent viral chromatin have hypoacetylated histones. Remember, when you methylate a histone, you repress. So they have hypoacetylated histones.
Hypomethylated, you mean?
Sorry. I'm not methylated exactly. Di or trimethylation. I'm reading the wrong part of the paper here. Yes. So the latent chromatin is hypoacetylated, which we just talked about, and it's methylated. And there's specific methylations on, histone H3 lysine 27, which we talked about last time. So also histone H3 lysine 9, specific patterns. So latent chromatin, not a lot of acetyl groups and methyl groups present. Okay? Now, the little twist here, there's a lot of twists in this paper. So it turns out that laboratory and clinical isolates are different. So in a high-passage laboratory strain, which has gone through cells for years, and this is called AD169, Alan Dove 169.
Right.
Right, Alan?
That's right.
The DAX defense is the only known transcriptional silencer of this IE1, immediate early 1 gene. All right? in CD4 positive. These are hematopoietic progenitor cells. But clinical strains have another restriction. Okay? And this has previously been shown to be another protein, a viral protein called UL138. And that's, of course, the protein in the title of this paper.
And that protein, the gene for that is missing from the laboratory strain that's been passaged in the lab for ages.
Exactly.
Which I guess you could think of as another domesticated virus.
Yeah.
So the passage that generates the difference is huge chunks of DNA. The clinical strains have or the passage strains have lost big chunks of DNA that are present in the clinical strains. So they are missing genes.
Probably because they've been passaged repeatedly in an environment where those genes weren't necessary.
That's right.
Exactly.
You know, it's interesting because in the old days, we all worked on pretty much laboratory-passed viruses. Very few people worked on new clinical isolates. And now more and more in the last 20 years or so, we've been doing this. And you realize they're different. This is a great example of that.
And am I correct that the clinical strains are in culture better at establishing latency in appropriate cells than the passage strains? Is that correct?
I believe that's what they said. Yeah,
it would make sense to me because the passaging is you're selecting artificially for viruses that will go through a lytic infection.
Right. So this paper is all about figuring out what UL138 is doing, right? So remember, we had this other protein, PP71, the viral protein that can mess with DAX. Now, this is a different one, UL138, and they say that the function of UL138 to silencing transcription during latency is not affected by this inhibitor of deacetylases, VPA, valproic acid. So they figured, well, it must be affecting histone methylation. So much of this paper looks at histone methylation. Okay, so UL138 function not affected by valproic acid, which targets the deacetylases. So we're going to focus on histone methylation. And I'm going to try and give an overview because there's a lot of data in this paper. You guys want to kick in stuff, go ahead. So the one experiment which I really like to start off, they take their laboratory strain, which is Allendove 169, and they put the gene for UL138 into it, right? They put an epitope tag on it, a hemagglutinin tag, so they can monitor its synthesis. And this virus, it grows, and it doesn't have a defect of any sort, but that virus, both viruses can establish latency, and valproic acid prevents the silencing of Allendove, which we know to be the case, but it does not permit silencing of IE1 transcripts when you add the AD138 in, when you add the UL138 gene into it. All right, so that's a really nice experiment, right?
Right.
Mm-hmm.
What were you going to say, Rich?
That says that UL138 is working on a different pathway than the DAX pathway.
So, and they've pulled UL138 out of the clinical strain, so any other gene differences you don't have to deal with. Right? It's just this one because, there are lots and lots of genes here, and you could argue that, well, maybe it's something else in the clinical strain. So there's a lovely experiment where you put it into the laboratory strain. You show it makes it valproic acid non-responsive, basically, right? So these results they get in both. They use these cells, THP1 cells, which I think are kind of monocyte macrophage lineage cells. We use these in the lab. They use these as a model for latent infections in their laboratory, and they also use embryonic stem cells, and they get the same result that this gene, the addition of UL138, makes the virus not responsive to valproic acid in terms of inactivating deacetylases. Okay. Is that good so far?
Mm-hmm. Mm-hmm.
Yeah, so then the next few paragraphs are all dealing with THP1 and embryonic stem cells, but I've already told you the answer to that. Then they delete UL138 out of AD138HA just to show that you absolutely need 138 for this HDAC-independent establishment of latency. Okay? So you put the gene in just to make sure you haven't done anything else. You take it back out and you show that it now confers valproic acid sensitivity to latency. All right. Now, they have a clinical strain that lacks UL-138. And this strain, if you add valproic acid in CD34 positive cells, transcription is silenced in the presence of VPA.
Right.
Correct. VPA does not activate immediate early transcription. So that says,
go ahead.
That says that UL-138 is not the whole story.
Right.
It's redundant
not the only
of something else. Okay, that's in the clinical strain that can do the same thing.
Mm-hmm.
They don't end up identifying it. They try. They go through a part of the genome, but they don't find it. They say basically it's one or more of the 200 different genes in the virus that's identified. We don't know what it is, but there's something else other than...
That's the next student's thesis.
That's right, other than UL-138. Okay. Now, let's get into the mechanism here. What's going on? Remember, this UL-138 is HDAC independent. So UL-138 can antagonize in an HDAC independent way. So they said, it must be histone methylation. So let's take a look at this. So they said, what does UL-138 do to the known repressive histone modifications at this major, i.e. immediate early promoter? And they found that there are repressive marks on the promoter when you infect cells with this AD138 virus, but much less when you infect with the wild-type virus. So this is already suggesting that 138 is having some effect on histone modification.
Good.
Because one of these methylations is present in the major promoter of AD169, and both in THP and CD34 positive cells, then it's, and this is lovely, they say UL-138 cannot be required for its writing. So the modification, the putting of the modification, the methylation, it doesn't require UL-138. And they think more likely it's UL-138 is inhibiting the removal by lysine demethylases. And so these are called KDMs, lysine demethylases, which I don't know if they've defined it in the paper, but if you're just reading this, they're full of abbreviations. It can be a little daunting.
No, they defined it, and the K comes from the single amino acid code letter for lysine.
Yes.
Okay. Now, they have different histone demethylases are known, and they have different names. One of them is JMJD3, and they found that, and they have antibodies against all of these. So they want to know which of these demethylases are recruited to the immediate early promoter, which would then be involved in taking off the methyl group. So they show that this one demethylase, JMJD3, is recruited to the major immediate early promoter in cells that is from the AD169 virus when you treat with valproic acid, showing that, and in fact they don't need viral transcription for that at all. When they add in UL-138 to the system, it decreases the association of this demethylase JMJD3 with the promoter, both in THP1 and CD34 positive cells.
Stem cells.
I need a glass of water, but I don't have any more left. It's all gone. So UL-138 is decreasing the demethylase's ability to associate with the major late promoter. So it can't take off methyls from the histones, remember? And the methyls are repressive, so it can't take them off to turn on transcription.
So it inhibits the removal of the methyls.
Right. You okay, Rich?
This hurts my head, but I've been through it a couple of times now, so I can get it.
So here's a lovely sentence, which I'll read because they put it perfectly. If inhibiting the lysine demethylase association with the promoter is the mechanism of action of UL-138, All right, so UL-138 is suppressing transcription. Then an inhibitor of the demethylase should have the same effect as UL-138.
Yes.
And that's, in fact, what they
That's
found.
what they find.
They have an inhibitor of this lysine demethylase, and they find that it prevents VPA from activating the immediate early mRNA. So basically, they think that UL-138 is silencing expression in latency by preventing lysine demethylases to be recruited to the major promoter. It maintains the repressive histone methylation, and therefore it's latent.
Right.
Now,
And then figure 5, by the way, is
a nice summary.
somewhat useful, except that it would have helped if they'd used slightly better labels, as Kathy pointed out. But if you stare at it long enough, it starts to make sense.
So it's a nice picture shows you the viral genome, the major immediate early protein promoter, and then it shows the DNA wrapped up over histones, and then acetylations and methyl groups.
Right.
And it's showing, for example, how DAX protein, which is the cellular intrinsic defense, is recruiting the acetylase to the chromatin to pull off the acetyl groups. Remember, acetyl off, transcription off. And then it's also showing how the UL-138 is interfering with the histone demethylases to keep it latent. Now, the last thing that I want to mention is that it turns out there is a cellular protein called CTBP1 that can modify the function of these lysine demethylases. And this protein, the cellular protein, can associate with the major immediate early promoter, the viral promoter. All right, and they have an inhibitor of this protein called MTOB. It's a product of the methionine salvage pathway, and when they treat THP1 cells with this inhibitor of CTBP1, which is a protein that can modify lysine demethylases, it inhibits association with the promoter. So MTOP inhibits CTBP1 association with the promoter, but it also inhibits the ability of VPA to rescue immediate early transcription in THP1 cells and CD34 positive cells. So CTBP1 acts sort of like UL-138, except that it's a cellular protein, which I found really interesting.
So one of the things that we haven't talked about that I find most interesting about all of this is that all of this has to do with modifications to the histones that are bound to DNA, either acetylations or methylations, around the major immediate early promoter, with the ultimate effect of shutting it down and shunting the infection towards latency in these monocyte type cells and stuff.
Right.
All part of it modulated by UL-138. UL-138 localizes to the Golgi. Okay? So all of these things happening in the nucleus at a very specific site are somehow being influenced by, or some of the things happening in the nucleus, the methylation part of it, are being influenced by a protein that is localized to the Golgi.
In fact, the cellular protein CTBP1 is also a Golgi localizer, and that has similar functions as UL-138.
Unfortunately, in their cartoon, they show it in the nucleus.
Yeah, they do.
So,
yes.
So that you're right. We don't know how that works, right?
Right. So there's a dotted arrow there.
Right.
Yes.
Okay, because we don't know how it's exerting the effect on the nucleus from the Golgi.
It's the network.
Right.
So UL-138 is blocking the demethylases from demethylating the chromatin, maintaining it in a latent state, essentially.
Right.
Now, these demethylases would, if it weren't for UL-138, the demethylases would be able to prevent the virus from establishing latency.
Correct.
Right?
Yeah. Taking off methyl groups will turn it on, right?
Yeah. Interesting to me, this is I'm getting anthropomorphic here again. Okay. It would, if you think of this as the cells trying to prevent latency, the consequence of that is to, since the virus is already there, promote lytic infection.
Right.
And so, you'd wind up making more virus and the cell dies.
You would in the cell.
Apparently, on an evolutionary scale, well, you don't necessarily make more virus, I suppose, but at least you go down the lytic pathway.
Yes.
So apparently, if there have been mechanisms evolved to try and prevent latency, they're done in the face of a consequence of a lytic infection. So I guess preventing latency is preferable to a lytic infection.
Well, but remember that these cells, as part of a multicellular organism, the selective pressure has strongly favored the cell, has strongly favored the organism's interests over the cell's interests.
Sure, absolutely.
So this cell has evolved mechanisms that are aimed at preserving the whole organism.
Exactly.
Which means it would behoove that system to have only lytic infections because those are the kind that the rest of the immune system can respond to.
Right. Okay.
Whereas a latent infection allows the virus to hide from the immune system, and so preventing the establishment of latency would be against the interests of the virus and in favor of the interests of the organism. Yes, it blows up the cell, but in this case, that would be a good thing.
Yeah, you blow up the cell, but then that's it. Presumably you don't have latency. What's interesting is that, so the virus comes in, say on the first infection, and latency is a good thing to be established so that the virus is now around forever. The really interesting question is then, how does it get reactivated? That was something we talked about in the herpes paper, right? What are the stimuli that reactivate it? I assume it also involves acetylation and methylation as well. But what is triggering it? We don't know it as well as for herpes simplex type 1 for sure.
I just want to remind people that there's the work from Eric Barton and Skip Virgin that herpes virus latency seems to confer protection from bacterial infection.
Right.
So latency may not be all bad. So again, if you're anthropomorphizing, what the virus wants to do, does it want to be lytic? Does it want to be latent? It's somehow perhaps dealt with being latent.
Yeah, if there's some host selection that keeps it too, that changes the whole landscape as well. In fact, we have an email about that later. But it could be that if it does confer protection or some benefit to the host, that would favor it. Yeah.
But at least in looking at this particular subsystem, it favors the story that these authors are trying to put forward, which is that this is a representative of an arms race happening at the level of latency.
Yeah, for sure.
So that the lysine demethylases would normally prevent the virus from establishing latency. The clinical isolate of the virus has this UL138 protein that prevents them from preventing latency and allows it to establish latency.
So I guess that when, let's say we have a clinical isolate, it's going into a myeloid lineage cell, right?
Right.
Undifferentiated. So the cell will just, this foreign DNA, the cell will just methylate the histone, it will chromatinize it and be methylated. Is that sort of the default occurrence on incoming DNA? Does anyone know?
Well, according to my little cheat sheet here, if I were the cell, let's get really anthropomorphic here.
You'd shut it down, right?
I would want to shut it down, and I would do that by putting on repressive methylations and avoiding acetylating it.
Right.
That's
because the acetylation would activate it. That would be my strategy. And yes, I would want to shut it down. But in this particular case, that's not, that works to the virus's advantage because it can establish latency.
Right.
Okay. So that sets off this war.
Obviously there's some conditions where demethylase are at play in cells, right? Mm-hmm. Because this UL138 is an antagonist of the demethylase. Right. So under what conditions is the cell looking at this, it's not integrated into the nuclear genome, it's episomal, it's chromatinized. what would cause the cell to look at this and say, we're taking those methyl groups off there? Or is it just if it doesn't have a certain protein bound to it, it gets demethylated? I don't know.
I know.
I guess that's the real question, is what's controlling the right. Because I know in cellular genes, some are maintained off and on by methylation, right? And there's certain signals for that. And what's going on here particularly? Because we don't have any, aside from viral genomes, there are no extra chromosomal pieces of DNA. Is that right? Unless you're infected with a herpes virus or some other virus. So maybe the cell can't make anything of it. So it's a little complicated, but it's basically down to controlling gene expression by acetylation and methylation, which is really a big deal. And as you said, Rich, for herpes, this came out of the Bloom lab, right?
Yeah,
the methylation.
I've been corresponding with Dave while this has been going on, and in fact, it's Nicole Kubat was his graduate student, and it was her idea originally, period, to look at chromatin modifications as perhaps playing a role in herpes latency.
So was this methylation or both methylation and acetylation?
It was methylation. Let me see here. We had just gotten the result with HSV DNA, we had just gotten the result that HSV DNA was not at all methylated. So we're looking for, so before we started to get into chromatin modifications as for controlling gene expression, in the era before that, it was all about direct methylation of the DNA, right?
Yep.
And if you want to know what controls latency and it's transcriptional at some point, you might look at methylation of the DNA directly, not the chromatin. So we just gotten the result that HSV DNA was not at all methylated. And she, that is Nikki, had just seen Alice's ALLIS, so an author, histone code paper, and wanted to try to adapt CHIP, which was a new technique at the time, chromosome immunoprecipitation, to mouse ganglia. I told her it would be a hard slug, and it is, because we're talking about now taking ganglia out of mice and doing CHIP. It really is tough, rather than doing cultured cells. But Nikki stuck with it for 2 years and really worked out the conditions, controls, etc. There you go.
So, in fact, CHIP was part of this paper. I didn't mention it, but that's the technique they used to see if UL138 protein interferes with the demethylase recruitment to the promoter. So they can chop up the chromatin, immunoprecipitate with an antibody to these demethylase proteins, and then say, what's the effect on the amount of the protein bound with and without UL138? Really cool technology. So this first letter is actually dealing with this same thing. It's a good segue. It's from Chris, who writes, Dear TWiV team, first of all, a very happy new year to you all. Thanks for your discussion of the paper on reactivation of herpes simplex virus from latency. It's great that we are getting a better understanding of the mechanism. The discussion reminded me of lysogeny, where temperate bacteriophage is latent in its host bacterial cell. In this case, latency is maintained by a repressor protein. When under stress of DNA damage, the host bacterial DNA repair mechanism kicks in. This also has the effect of degrading the phage repressor protein, allowing the phage to switch into lytic mode with the production of progeny phages. It is always tempting to view viruses as clever little beasties being able to respond to changes in their host cells and enabling them to escape from a potentially damaged cell, but this isn't the case. Vincent said that, quote, if the neuron is going to die, the virus needs to replicate and get out, unquote, end quote. But why? Why do viruses and neurons need to replicate and get out? Viruses have no desire to survive. They survive because they possess features enabling them to continue infecting and persisting in their hosts. Viruses, like all other infectious agents, are populations, not just individuals. Individual viruses do not adapt or evolve an ability to respond to changes in their host cells, enabling them to escape and survive, but populations of viruses certainly do evolve. It is interesting to ask why HSV or phages can respond to stresses in their host cells by switching into lytic mode, but all we can say is that somehow the ability bestows a survival advantage on the virus. After all, if my understanding is correct, latency and reactivation from latency is a feature of all herpes viruses, and therefore the ability to reactivate is unlikely to be accidental. regarding the discussion on inhibiting reactivation. An ability to prevent symptoms resulting from reactivation would be great, but how would we know when to administer an inhibitor, even if we had one that lacked significant side effects? But at least we do have a number of effective antivirals against herpes viruses and a vaccine to prevent reactivation of varicella zoster virus in neurons of the elderly. Well, to that last point, I would say that if you're going to have an organ transplant, right, and you don't want to reactivate, then that would be a case where you give people these inhibitors if
Right.
we had ones, right? No, I agree with this point. You know, that's what the problem is with anthropomorphizing. We're saying, well, the virus has to get out of a dying cell, but maybe not. He's absolutely right. It doesn't matter because the population as a whole has this property of latency and reactivation, and that will enable it to survive.
But when we say the virus needs to get out of a dying cell, I take that as shorthand for there has been selective pressure over the virus's evolutionary lifespan that favored viruses that got out of cells before they died.
That makes better sense. Yes,
Yeah,
I like that. Yeah,
And so the shorthand for it is anthropomorphizing, but what is actually meant when it's a scientist saying this is this whole, convoluted block of text, which gets at the point that this is.
You see, Alan Dove, why anthropomorphizing is bad. It can change the meaning of something.
Right, but you see why it's useful?
No, I like talking.
Yes.
I understand why it's useful.
Yeah.
I know that. But anyway, that's a great letter. I thank that. Chris is at Middlesex University. He is a senior lecturer in microbiology there.
Cool.
Kathy, can you take the next one?
Sure. Jim writes, Vincent ran viruses through Ngram and noted the percentage of references fell off in recent years. I'd heard that published titles in 2014 were 400,000, but in 2015 they jumped to 700,000. Those numbers aren't defined, but do indicate published material has greatly increased. It makes me wonder if the percentage of references of virus fell off because of the huge number of increased publications, but the actual number could have increased. It would be helpful if Ngram could provide raw numbers too, because references to viruses may be steadily increasing but are overshadowed by the increasing amount of material that's available. Interesting site to play with, though. Thanks. And that's from our friend Jim in Smithfield. And so I don't know, maybe we'll hear more from, it's Anthony, right? About that issue, percentage versus absolute numbers. And just to point out, for people who maybe have never gone to the website or tried this, it sounds like Ngrams, EN, but it's spelled capital N-G-R-A-M-S when you try and find the site in Google.
So by Anthony, were you referring to the person who developed
said,
this?
Thank you. Yeah. Right.
At Google, the guy who developed it at Google, that would be that's Fernando.
Fernando, that's right. Sorry. Yep. Okay.
Alan, can you take the next one, please?
Sure. Mark writes, Hello, Vincent and the TWiV-osphere. Happy New Year 2016. Congratulations on launching Microbe TV. In episode 369, Vincent mentioned sampling a physics-related podcast and that he didn't want to regularly listen because of its poor audio quality. As I recall, only Alan Dove agreed, commenting that poor audio quality is inexcusable. Since 2008, I've sponsored over 50 different podcast shows for several high-tech companies. The first thing I looked for is consistent high-quality audio. The second factor was the level of engagement between the show's host or hosts and its listeners. Podcasts, like old-fashioned radio shows, are an intimate medium where listeners voluntarily give their time and attention to the show, its hosts, and guests. Listeners will consider, and I dare say trust, recommendations for products that hosts genuinely use. Due to the intimacy of the podcast medium, content quality does not compensate for bad audio quality. Bad audio quality is an insult to listeners' ears and an affront to their intelligence. Sadly, some podcast shows never improved their audio to the level where I would sponsor them. TWiV listeners who aspire to create podcasts take note. Borrow and adopt how TWiV engages listeners, and copy or improve on tools and techniques for producing consistent high-quality audio. Here is a Shakespearean analogy from Hamlet in Act 1, Scene 3. Polonius shares worldly wisdom with his children, Laertes and Ophelia. One line applies here. Clothes don't make the man, they announce him. So it is with podcast audio quality. Enough of this gushing appreciation. Oh, no. Here are some
more.
Yes. Here are some show ideas. Repeat visits by Gabriel Concerto in B. Victoria, Lynn Enquist, Carla Kierkegaard, or Eugene Koonin. A stretch goal is to book and interview Nobel laureate James D. Watson as a special TWiV guest while time permits. My father had a mild acquaintance with Watson, whom I met briefly when he came to express condolences in my dad's wake. Now for some fun. About 18 months ago, I sent you a bingo card based on common phrases used in your podcasts. In the interim, there has been phylogenetic drift in the phrases used in the TWiV-osphere. Attached are updated bingo cards reflecting my observations of changed phrasing. Bingo squares containing gain of function or the name Dixon are the most obvious changes since the last TWiV bingo card was drafted. Today's weather in San Jose, California, after 4 days of rain. The El Nino weather pattern is opening up, and we're finally getting good rainfall in this drought-stricken region. Let's hope things continue for the next few months. With luck, a precipitation-heavy weather front will pass from California eastward and deposit on Michigan, Massachusetts, and New York. All right.
The rain can stay in California if you want.
Yes, that's fine. We've got plenty of it here in Massachusetts.
It's a nice bingo board.
It's a good.
It's got a new bingo card with a bunch of new
I've been corrected by Jane. Cherry blossoms, I think, are new.
Yeah.
Gain of function appears twice.
I like be nice to Dixon.
Be nice to Dixon. Yes.
Did you read the paper, Dixon? It's another one. Although I don't think we ever say that to him exactly.
Oh, yeah.
Oh, yeah.
We do?
Oh,
we do?
Yeah. Oh, yeah. I don't.
You
You do.
Oh, it's all my fault. Yeah, I agree. It's my fault. I wish he would read the papers.
Yeah. We have a show title.
We have a show.
All right.
Long list of authors.
So Mark works for Drobo, who sponsored us many years ago. He sponsored us in the very beginning, and we appreciate that. That really helped us to get going, and he obviously liked us.
And I still have my Drobo sitting on my desk, humming away, storing backups of my files.
Wonderful.
Nice.
Wonderful. That was before you joined, Kathy.
Mm-hmm.
Maybe even you as well, Rich.
No, I was here with Drobo.
You were here, and we appreciate that. And Mark,
we had a Drobo giveaway contest.
Yep. Mark is a long-time listener. And I remember at an ASV someone coming up to me and saying, I just want to let you know I won one of the Drobos. We should do it again, Mark.
Yes. Yeah. Well, I think Mark is not with Drobo anymore. Wasn't that what happened?
He left, but then he came back, is my understanding.
Ah.
Yep. All right. Rich, did you read one yet? No, you haven't.
No. Bob writes. Good morning and Happy New Year. I just heard your picks on the New Year's Eve show and wondered if you've come across Make Magazine and the whole maker movement. This is apropos of the Edmund Scientific Catalog and Heathkit. If you hadn't seen it, check it out. And he gives a link to makezine.com. There's a whole ecosystem growing up around home technology development. And I had a look at this, and it's pretty amazing.
Alan,
I'm
did
one
you
of the
pick
originals. Yeah, I'm one of the original subscribers to Make. I subscribed to them from issue 1.
I think you had it as a pick long time
I think I may
ago,
have
right?
picked it, yes.
Yeah.
I also came across this today on a veterinary newsletter I happen to subscribe to. And it's an article about travel-related Zika virus infection has been identified in the Harris County area in Harris County, Texas. The Harris County Public Health and Environmental Services has received information from the CDC that Zika virus has been confirmed in a traveler who recently returned from Latin America. The individual developed symptoms that are associated with Zika virus, which include fever, rash, and joint pain. It's clear and cold, 10 degrees Fahrenheit today in Chicagoland after snow last night. From Bob. And we have a note here. Is this from you, Kathy?
Right. Maren McKenna had reported in her blog that there was a case in Puerto Rico in someone who hadn't traveled. And so if you count Puerto Rico as a U.S. territory, then it's in the United States.
Which is not too surprising, because if it's in Latin America and going into the Caribbean, then that is a huge hub for the Caribbean.
Mm-hmm.
And we've got the vector, right?
Oh, yeah.
It's 80s?
Yeah.
80s?
Yes, we do.
So,
yeah, this with more infections locally around us, they will come here, just like dengue and chick. Sure,
right.
And there was even news this morning on the radio that CDC is considering, or some federal agency is considering recommending pregnant women not travel to some of these South American locations.
Right.
You listen to the radio, Kathy?
Yes, I do.
Where in the car? You don't drive though, right?
Oh, at home before I leave.
Really?
Yeah.
Wow. That's very interesting. Is it a tube radio?
No, it's clock radios all over the house.
I'm giving you a hard time.
Yes.
The next one's from Jarrett. Hello, virologist. This morning I read a report that a confirmed case of Zika virus has turned up in Harris County, Texas, rather near to me in Austin. Having listened to TWiV 368, my ears perked up at the mention of said flavivirus. Often such illnesses are acquired by tourists traveling abroad who become symptomatic after returning to the U.S., whereupon the discovery is made that we have a new viral neighbor. My question is this: what is the time frame wherein an individual infected with a mosquito-borne flavivirus could transmit the virus to another mosquito of the appropriate genus, Aedes, who may then go on to possibly spread the infection? Is it possible for a latent flavy to reactivate once it's in the body of an Aedes mosquito? In other words, once a person has recovered from their infection, could they spread the virus in future exposures to mosquitoes? And more generally, is it so that all viruses have periods of latency? These questions came to me as I wondered if the infected person in Harris County was asked to avoid the outdoors in order to prevent further spread of Zika, or if such a measure would even be necessary. As always, thanks for the excellent podcast. Right now, it is a very comfortable 16C in cloudless, Zika-free Austin, Texas. All right, so the first question is, what is the time frame wherein an individual infected with a mosquito-borne flavy could transmit the virus to another mosquito of the appropriate species? Okay, well, I don't think we know for Zika, but, the incubation period before symptoms is 3 to 12 days.
That's right.
So you can be incubating virus before you know it is in you, and so if the amount of virus in you is high enough, then a mosquito could bite you and pick it up. But that's the key; it has to be high enough infection level, and if not, it won't spread to others. So, and remember, the likelihood that you're going to get bitten is not huge in Texas, I would guess, particularly at this time of year. So, it's unless you have lots of Zika infected people, it's not going to happen. I don't know of any latency in the flaviviruses.
No, neither do I.
No.
Not all viruses establish latency.
Yeah, these are acute infections. The virus comes in, it replicates, there's a defined period of time, and then the host either dies or it's cleared.
Right. The underlying theme of all these questions is, could Zika virus become established here? The answer is yes, but it requires a number of infected people and plenty of mosquitoes. We have plenty of mosquitoes. We don't have a whole lot of human exposure to mosquitoes at the levels that you see in Central and South America.
Right.
So in Austin, in the summertime, when it's mosquito season, people are running from their air-conditioned SUVs into the air-conditioned shopping malls and back to their air-conditioned homes, and you're not going to have the kinds of exposures that you get that really sustain an outbreak. But you can. We've got chikungunya occasionally, we've got dengue occasionally in the southeast and such. So, yeah.
I remember West Nile wasn't here.
West Nile
was introduced into New York City.
I was thinking about that, but the interesting thing about West Nile is that it infects birds.
Right.
Correct.
They're a primary reservoir. So
correct. That probably helped a lot, right?
And they don't go running inside to the air conditioner.
Right.
Okay, so that probably did a huge amount to amplify.
Makes a big difference.
Amplify the West Nile, which of course spread from the East Coast to the West Coast in a couple of years. It was like a wildfire.
Right.
So does
Zika et al. Are only in primates, as far as I know.
As far as we know, yeah. So if Zika were in birds, it would be a problem. It could spread more extensively. Same with dengue, right? Dengue does not infect birds, and therefore it's harder for it to gain a hold in, say, the U.S. and so forth. Interesting. All right, we only have two more. Should we finish them?
Sure.
Kathy's next.
So this is Bob writes. Now, is this the same Bob as the one a moment ago?
I don't think so.
Yeah. Okay, just different. Okay. Dear TWiV-ola, regarding your discussion on temperature conversion, when I was a kid, my dad taught me an approximate conversion you can easily do in your head. Take the temperature in C, add 17, double it, and subtract 10%. This gets the right scaling, but the result is offset by a degree or two. Partly sunny and 33 outside Chicago today, with humidity 61% with 16-mile-per-hour wind. Bob. Yeah, that's
never-
about like,
Yeah.
take the temperature in C, multiply by 5/9 or 9/5, add
yes.
or subtract 212.
Not much better than the official formula.
I mean, if you're good at arithmetic in your head, fine, you could do the whole thing. I just like a few numbers, like 16 is 61, 28 is 82.
Right.
Yeah, so.
And the last one's from Paul Allen.
Paul writes, not Paul Allen.
How do you know?
But maybe, hey, we could use some sponsorship. Paul writes, Dear TWiV, thank you for your interesting discussion of our manuscript on biofilms and filamentous phage in your recent podcast, TWiV 368. Definitely not Paul Allen. To answer your question, we haven't looked at Phi-X. However, since its structure isn't filamentous, I don't think that it will assemble polymers into a liquid crystal. It's a good question, though, and I'm constantly surprised by what phages can do. We'll check and let you know. As for the question of what's in it for the phage, perhaps by making themselves useful to their bacterial hosts, PF phages have managed to increase their abundance and prevalence. Certainly, they seem to be ubiquitous among Pseudomonas clinical isolates and are present at very high concentrations, averaging 10 to the 7th per milliliter in Pseudomonas biofilms. Pat and I like the idea that PF phages may be symbiotic with their Pseudomonas hosts. In support of that argument, PF phage production is under host regulatory control, and these phages are, for the most part, non-lytic.
Cool.
So Paul is the senior author on that paper that we did, and I made fun of the fact that he would probably never listen, so I have to I have to eat crow, and along with it, West Nile virus.
Yeah, there you go.
Yeah, and I'm happy for the fact that he speculated on what's in it for the phage, because that was my concern.
Yeah.
What's in it for the phage? Well, Chris would say nothing's in it for the phage, maybe for the population. Alan, do you have a pick of the week?
I do have a pick of the week. So this is something that I just heard about as part of a conference that I'm writing up. When I just realized I linked to the sign-up part of the site, I'm going to
you could change
fix that. So this is it's part of a study. Trying to fix the link while talking. So there we go, and the link is fixed. These folks took the idea that there are a lot of viruses out there, and they want to sample the virome. And the way they're doing it is by getting volunteers in, I think it's just in New York and Massachusetts. But the volunteers sign up on the website. They then receive a kit that has a couple of sterile tubes and swabs in it. If you and also a feces sample cup if you volunteer for that part of the study. And if you develop a cold or you think you're coming down with the flu or just it's time to take a sample, you do a nasal swab and I think a sputum sample, and you send it in, and they do deep sequencing on it and find what viruses are in your sample. And they are collecting from this maps of the viromes of cities, which I think is a really cool project.
Yeah.
So
who is they, and how are they funding this?
Is
Bhumi Chinara, and it's National Science Foundation, which I find very surprising, because I always thought National Science Foundation doesn't do anything with pathogenesis or disease. So,
right. So that's the crew behind it, and I don't know what the basis of the grant was, but. You could argue that this isn't really a pathogenesis or disease study. It's a microbiome of the built environment study.
Yeah. I mean, somehow NSF funded.
However,
so
they put
great.
it in the grant, it worked. So there you go.
Yeah.
I just tried to sign up, and they said my email is already in their system.
Really?
I don't know how I never did this before. It's interesting. Well, I would love to participate. This is the same sample collector thingy that 23andMe uses.
Huh.
You spit into it, and then when you close the top, it releases a solution of SDS and something else that then preserves it on lysis cells. It's pretty cool.
So I signed up.
All right.
And I'll let you know how it goes.
All right.
So is it geographically limited?
Now, the talk was geographically limited, but that was a New York Academy of Sciences event. And so they focused on their results from New York and Massachusetts. And they talk about New York and Massachusetts in the, on the website.
You just try it.
Mention any other states. You should go ahead and try signing up.
I mean, people listening to TWiV are going to sign up, and they're going to get people from all over the place.
Mm-hmm.
And I'm sure there's some limit to the number of signups that they can fund, but
I think it's really great.
Yeah.
Kathy, what do you have?
I picked The Thing Explainer. So,
excellent.
Good. So Alan already knows about it. I got it for my birthday, and this is written by Randall Munroe, the author of a book, What If, and also the creator of XKCD. And this is a book that I think you have to get in book format. You can't read it on a Kindle.
Right.
And he explains complicated stuff in simple words. And so we've talked before about Upgoer 5 and the 1,000 words that people use the most. And he talks in the back about, he lists his words and the fact that there's lots of different ways you could come up with the 1,000 most used words. And he used mostly those, and then evidently in a couple of places he didn't. But all kinds of really cool things are described. The first one that I spent a lot of time on is the bending computer, which is a laptop which bends because the screen can fold down, and then it's got all the stuff on the inside of it. He just does all kinds of really cool things. But this one I want to read to you. So the whole page is called Tiny Bags of Water You're Made Of. Okay, so those are cells. And then a very tiny paragraph here with very tiny items. Things that make you sick. These tiny things can get into your bags and take control of them. When they do that, they use the bag to build more of them. When the kind shown here gets into you, your body gets hot, your legs hurt, and you have to lie down. Your whole body feels bad, and it makes you hate everything. You feel like you're going to die, but usually don't. We say all life is made of bags, but these things aren't. They also can't make more of themselves. They have to get a bag to make them. So we don't know if it makes sense to say they're alive. They're more like an idea that spreads itself. I highly recommend this book.
Yeah, my son had this at his house when we visited in Seattle a few weeks ago, and I had a look at it. Zeroed right in on the cells and the viruses. It's very cool.
Does he talk about repressive chromatin?
No.
Wonder how he'd do that one.
Right, but you know he's got hand computers, which is really a smartphone. It's got our star, things about a car engine. I forget what he calls batteries, something like power boxes. It's just really great, and it's just his really funny sketches sometimes, and just delightful. Cool.
The Upgoer is a Saturn V rocket, right?
Yes.
Something like that.
Upgoer.
Yes. And by the way, if you Google Upgoer 5 text editor, you get to a web page that will allow you to compose things in this style.
Weird.
So you can write something and it will check to see if you've used only the 1,000 most common words, and it'll let you know which ones you have to fix.
He also has a link. Where did I see that? Anyway, and then the very last page is this giant fold out, kind of Eloise style, if any of you read the Eloise books where she's got the Plaza Hotel. Anyway, it's called Sky Toucher, a look inside a really tall house. And so it's a skyscraper, and it's got all the features of a skyscraper, a tall apartment building. It's cool.
Nice.
Neat.
Not Dixon. Rich Condit.
Do you have a pick?
You have a pick, Rich Condit?
Yeah, I have a pick. I picked a Christmas present I got, which is a Galileo thermometer. I had been familiar with these before, but I finally got one, and it's sitting here on my desk. So for those of you who are not familiar with this, I gave the Wikipedia link to it. Oh. It is a tall sealed tube of water that has inside it smaller little sealed tubes that have colored fluid in them, probably ethanol, and attach to them rings that have numbers on them in the temperature range that their thermometer uses. And all of these little colored fluid containing balls inside the big cylinder have different densities. And so they will float or sink depending on the density of the water. And it's been all adjusted just right so that if a ball sinks, it is in excess of that temperature. And if a ball is floating, it is lower than that temperature. And if a ball is sort of suspended in the middle, it's probably right at that temperature. And it works like a charm. And it's very entertaining.
What's the temperature in your house right now?
That's a good question. Let me look at my Galileo thermometer. It is less than 68 degrees.
That's very helpful.
It is between 64 and 68. And the 68 looks like it could, kind of, if I kind of move things around here, it's on its way down. So it's probably about 68. Oh, there goes the 68. See, I just jostled it.
Mm-hmm. Yeah, those are cool. Yep. I have one as well. Someone sent it to me years ago, but they mailed it and one of the balls arrived shattered. So
bummer.
Kind of not accurate. That's why I use Celsius to this day.
It must, it must be sensing pretty small changes in the density, I would think. Yeah. I don't know. I haven't looked into it that deeply, but
very, very cool.
Pretty interesting.
My pick was something that Mark Martin shared on Twitter yesterday. Mark Martin is a professor of microbiology at the University of Puget Sound, who is very active on Twitter, and he sent out this article of people making popsicles from different items, but using 3D printers. And one of the items, of course, is a virus. Basically, they 3D print what they want to make, a virus. They did some cacti, a few other things. Then they make a mold around it and take it off and get rid of the 3D printed thing. And then you can pour in your sugar solution and let it freeze and put a popsicle stick in, and then you can take it out and eat it. I think this is cool. They're all different colors, and they have a movie of people eating them. They're called Dangerous Popsicles, Aesthetics of User Unfriendliness. And there's an article at a 3D printing site, which I'll link to. And then there's also the website where the creators have a website called Dangerous Popsicles, and they have a little video of how they did it and some photos. It's really cute.
These are great. Really cool.
It's just great. The virus ones are great.
Yeah.
And they're beautiful. They're very pretty.
Mm-hmm.
So thank you, Mark Martin. I give him total credit. He didn't submit it as a pick. He just put it out on Twitter. So I figure when people do that, I can steal it as a pick, right?
Sure.
It's no problem. So our listener pick came to Kathy. So why don't you tell us about that, Kathy?
Yeah, so Christiana sent this to me. She wrote, "This is a nice article that summarizes how to give good PowerPoint presentations. Nothing new in there, but it highlights the main points, so it might be good to spread the word widely." And it's a link to science, the science, is it the careers section? Yeah. Using PowerPoint to shine on stage. And so it's just a short question and answer format feature. And this is a way to segue into the fact that a long time ago, I picked Jean-Luc Dumont and some of his website and videos and things. He has been signed on to give the career education workshop at ASV in Blacksburg this summer. So the Tuesday afternoon at 4:30, before the banquet, that presentation time, he's going to be talking also about how to make really good PowerPoint slides, which should serve everybody well, because even no matter what you're going to do in the future, you're going to have to give presentations probably in some shape or form. And so this Hints in Science is something you can look at in the meantime, and then come to ASV and hear Jean-Luc Dumont.
I read this article. It's nice. This is a topic that just really gets me revved up, because so many people abuse PowerPoint and give just miserable talks. And I think the two major themes that are right on the button in this, right on the money in this article are minimize text and connect with your audience. Okay.
Exactly.
It used to be that in this sort of lecture format, what you did was you didn't have any visual aids. You stood up in front of people and you talked. And when I was in college many eons ago, the only visual aids that we had were chalk and a blackboard. Okay? So the instructor was communicating directly with the audience, and any material he put on, he wouldn't write text on a blackboard. Okay, you had a textbook that you could go to. There would be illustrations that the instructor might put down. And in fact, the pace of the communication of the information was dictated by how fast he could write an illustration on the board, which meant that it went by pretty slowly, which is good. So now we come up with handouts and overheads over time, and finally PowerPoint, where now you can illustrate things. It allows you to communicate much, much, much too much information, to provide much too much information in a short period of time. And I think the focus has to be on you are speaking with these people, and you're making eye contact with them, and you're communicating with other human beings. And this stuff on the board here is just a few simple things that are relevant, not fancy, irrelevant pictures, but relevant things to illustrate your points. Okay?
Yeah.
And so that's what I would do for the medical students, right? I'd give these lectures and I'd have, a table or an illustration to illustrate what I was talking to them. And there was constant pressure to I also give them an outline, okay, to put everything I was saying on the PowerPoint slide, okay, because they wanted basically a record of this. They didn't want to go to the lecture. They didn't want to listen to me talk, okay? They didn't want the communication, all right? What they wanted was something that they could hold on to and study from. Okay, I get it, but it compromises the quality of the actual presentation.
Right. This is also rampant in the business world, where it's become standard for people to distribute a PowerPoint presentation as if it was a text.
Right.
And they'll say, "Oh, well, here's a PowerPoint on that," and they'll
Right.
just send you a link, and it's slides that have the text written in the least efficient format possible, which is really, really perverse. But most of the business world has now standardized on this, and it has crept into science as well. And yes, this is a really, really unfortunate thing, and I'm very glad to see this is an excellent pick.
So, PowerPoint, I suppose, can be used in that fashion to be shipped around and communicate what you're
There are
talking
better tools for that, though.
Yeah, like
That's
real
done
text.
because people are too lazy to write something.
Yeah, okay.
Yeah,
Or
well,
because, or
it
because
certainly
they
doesn't
actually
make a
don't
good
have much
talk.
to say.
yeah, it comes into play in classes where, students are clamoring to have all the information that you're saying in lecture on the slides,
Exactly.
and it's because they don't know how to take notes anymore.
Right, that too.
So, they haven't had to take notes because they've been handed PowerPoint presentations.
It's frustrating for me because I enjoy speaking and I enjoy giving talks, and what I enjoy about it is the actual communicating with the audience, standing up and looking at people in the face, seeing their reactions, getting questions, actually having some sort of communication rather than being a robot. Okay, and when the pressure is on to put everything in the PowerPoint presentation, it's moving way away from that. And then since that is what so many people do, the students learn to do it that way. And so many of the talks.
Kathy, are you the one who just sent this text?
I am, yes.
Yes. Power corrupts. PowerPoint corrupts. Absolutely. Yeah. Excellent.
All right. Enough. That's my soapbox.
There we go.
Yeah, that's good. It's good stuff for sure. All right, that will do it for TWiV 372. You can find it at iTunes and also at microbe.tv/TWiV. We love to get your questions and comments. Please send them to twiv@microbe.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is in Gainesville, Florida. Thank you, Rich.
Sure enough. Always a good time.
And Alan Dove is at turbidplaque.com, also on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.